Thank you for this introduction. Thank you to accepting having me here, and I hope you all had a nice coffee break. Yesterday was the theme of troponin. This morning it's DPP3. I will present to you my work of DPP3 in cancer and chemotherapy-induced cardiotoxicity. It's the first part of my PhD project. A very quick reminder, you've seen these slides already. But DPP3 is an enzyme, mainly intracellular. It helps the cell produce antioxidative stress proteins, but it can also be found in the circulation as a CDPP3 will be called in this presentation. And my colleague Noma showed earlier these exact same graphs, but it seems to be predictive of poor outcome when high in acute critically ill patients. Something that hasn't been presented today yet is the role of DPP3 in cancer. Very recent papers showed that DPP3 is overexpressed in different types of cancer, and here it's a retrospective case control study on esophageal cancer that showed that when DPP3 is overexpressed in the tumor tissue, not in the circulation, in the tissue of the tumor, the outcome of the patient was much poorer, and these results were found in many other cancers. And therefore, the authors of these papers did in vitro experiments with depletion of DPP3 in cells, and it inhibited migration, proliferation, and it promoted apoptosis in colorectal and esophageal cancer models. And furthermore, they did in vivo models, and when suppressing the DPP3 expression in the tumor didn't grow at all, almost in the mice injected with the tumor, and the mice survived more. So the conclusion of this slide is that DPP3 seems to be like an oncogene, and it functions through a cyclin-dependent kinase pathway. The objective of this presentation is to answer to these two major questions. First of all, is DPP3 secreted by cancer cells and also maybe cardiovascular cells? And what does the effect of the inhibition of the enzymatic capacity of DPP3 on cancer cells and cardiovascular cells? And is it cancer type dependent, and is it condition dependent, stress condition dependent? So for that, we built a cell culture model where we had human cardiomyocytes, mouse ovarian cancer, mouse breast cancer, and we submitted them to stress in form of serum-deprived media or doxorubicin treatment, and we collected the culture media after 24 hours to measure if there were any DPP3 in the culture media. And we also inhibited DPP3 with our antibody protsizumab, and we studied the effect on cell viability. The second thing we did is a bit more complex. I hope you will understand, but we submitted the cancer cells to stress, and then we collected the culture media. And our theory is that there is a lot of DPP3 when the cancer cells are stressed, and we apply this media to human cardiomyocytes. So it would be a conditioned media. And after that, we measure the cell viability, and also we inhibit the potential DPP3 in this conditioned media with our antibody. Lastly, we have an in vivo model of doxorubicin-induced cardiomyopathy on wild-type and knockout DPP3 mice. And we inject them with doxorubicin for a week, and we measure the heart function, the DPP3 levels in the plasma for 3 weeks before sacrifice. So the first results here are quite interesting. Yes, the answer is DPP3 is released from cancer cells and cardiomyocytes. So in the middle here, you can see that on human cardiomyocytes, the more stressed the cells were, the more DPP3 you could find in the culture media. And it's not only mortality dependent. There's probably another mechanism to that, because, for example, in the middle of the overgraph on DMM, which is a serum-deprived media, you have more cell viability than on the doxorubicin treated cells, but you have more DPP3 in that culture media. So it's not only mortality dependent. These results were shown in the cancer cells lines we had too. Secondly, when we inhibited the DPP3 extracellularly with our antibody procizumab, we fully restored the human cardiomyocyte viability after 24 hours when submitted to stress by doxorubicin. And we didn't restore any viability on the breast cancer cells. We did slightly restore murine ovarian cancer cell viability, but not as fully as human cardiomyocytes. Thirdly, this was the more complex experiment that I was talking about. We submitted our cardiomyocytes first to our regular stress with serum-deprived media and doxorubicin. We had indeed an increased mortality. Then we applied secretum, so culture media from cancer cells that were not submitted to stress in any ways. And we didn't have any particular impact on cardiomyocyte viability. And thirdly, what we did was we applied the culture media from cancer cells that were stressed by doxorubicin, and we had a significant increase in mortality of human cardiomyocyte. And when we applied our antibody procizumab on this cell culture, we significantly restored the human cardiomyocyte viability. Lastly, the mice model with knockout mice for DPP3 showed that the knockout DPP3 mice are protected from the cardiotoxicity induced by doxorubicin, and also the wild-type mice that were submitted to doxorubicin had a significant increase in mortality after 3 weeks compared to the knockout mice. So all of this, as first part of my PhD project, shows that DPP3 is indeed released by cancer cells and cardiomyocytes, and it's not only correlated with the mortality of the cells. And CDPP3 seems to impact cardiomyocyte survival when inhibited. The question that is remained to be answered is which one is important, the one secreted from the cancer cells or the one secreted from the cardiomyocyte? And the questions we have to answer in the future now are how does DPP3 affect the cardiomyocyte physiologically? Which kind of stress leads to a lot of DPP3 secretion? And we have to build an in vivo model of mice, not only with doxorubicin or chemotherapy, but also with tumor, as it would mimic more clinical situation for our human patients. I would like to thank all my team in Paris, and thank you for your attention.
Excellent job. Questions from the audience?
I was very clear, apparently.
Thank you so much for your presentation, very interesting. I have a question. Is the DPP3 also released by myeloid cells or like inflammatory cells?
So from what I know, the DPP3 is expressed in every cell type. Mm-hmm. Then if it's secreted by every cell type, I don't know. If we say that the secretion is mainly due to apoptosis or cell death, well then probably, yeah, it could be, yeah. And we're also going to, I didn't show this here, but we are also going to analyze on endothelial cells this whole protocol to add more value of like to represent a more human heart.
Fantastic. I was just saying that if you want to really dissect or exclude the secretion of DPP3 from myeloid cells or bone marrow cells, you just can do the bone marrow transplantation in mice and then subject to your injury. That will exclude all the inflammatory cells.
Okay, yeah, okay.
Thank you very much.
Thank you.